Okay, so the next session is going to be on microbiomes, health, and the built environment. I'm Rob Knight. I'm the director of the Center for Microbiome Innovation at UC San Diego, and I'll be kicking the session off. In addition to me, you'll be hearing from Erica von Mutius, who is one of the leading experts on the hygiene hypothesis and why perhaps your child should lick a cow to be healthy. After that, you'll hear from Jessica Green, who's one of the leaders in understanding the microbiome in the built environment. And then finally from Jack Gilbert, who's going to tell you about the microbes in situations ranging from your homes to your hospitals. So just to kick this off, as you're all familiar with, sequencing the human genome was a remarkable investment, and so about $3.8 billion was spent on achieving the human genome sequence. And as Obama mentioned in his introduction of the BRAIN Initiative, every dollar that was invested in that yielded $140 in return. So it's been an incredibly prudent and incredibly rewarding investment. However, when we think about our genome, what's in it is about 20,000 human genes, more or less, depending on what exactly we count. But the size of our microbial gene catalog ranges from 2 to 20 million microbial genes. So in terms of gene count, we may be less than 1% human. And just like we leave our human DNA on everything we touch in ways that can be used for forensics, the same is true for our microbial DNA. And Jack's going to talk more about this in his talk, but a few years ago with Noah Ferrer's group at Boulder, we were able to show that we could match the palm of your hand up to the computer mouse you use with over 90% accuracy, and the tips of your fingers to the keyboard that you type on. And this came out in PNAS back in 2010, but more importantly, it was on CSI Miami, so you really know it's true. But all of this raises the important question: is it really okay to focus on the human genome and ignore 99% of the genes that are in our bodies? You might have a few qualms about that, especially given all the traits that are now being linked to the microbiome. And these include all kinds of things that you might expect, like inflammatory bowel disease and irritable bowel syndrome, ones that are a bit more surprising to link to your gut microbiome, like cardiovascular disease and colon cancer. And then things that are really surprising. So, for example, Dan Littman at NYU has shown that you can link the gut microbiome to rheumatoid arthritis. And one thing I've been working on with Jeff Gordon's lab for about the last decade is microbiomes and obesity. And today, for example, I can tell you with 90% accuracy if you're lean or obese solely by looking at the DNA of the bacteria in your gut. So on the one hand, this is kind of a cool technical trick. On the other hand, we don't think it has a lot of commercial potential as a test for obesity, because you can probably tell which of these people is obese knowing absolutely nothing about their microbes. But on the other hand, if you have every gene in their genome, you can only do that classification task, lean or obese, with 58% accuracy based on the human genome versus 90% accuracy based on the microbes. And this is amazing because it suggests that those 3 pounds of microbes each of us carries around with us can be more important for some of these traits than every gene in the human genome. Now, fascinatingly, over the 20th century, the incidence of a whole lot of infectious diseases, ranging from measles to tuberculosis, declined precipitously. But in the same time, autoimmune diseases, ranging from multiple sclerosis to Crohn's disease to type 1 diabetes to asthma, all skyrocketed over the same period. And one thing that's interesting about this is that when this paper was published in the New England Journal in 2002, none of those autoimmune diseases had been linked to the microbiome. But today we know that all of them have microbiome involvement. And so one fascinating question is whether this focus on controlling single pathogen species has led to a microbial silent spring situation in our gut, where loss of diversity may be contributing to some of these disorders. And this is something that Marty Blaser has explored at book length, particularly with reference to antibiotics, but also with reference to C-section, to hygiene practices, and to all kinds of other things that separate us from the healthy microbes in our environment. One thing that's interesting to consider is many of the chronic diseases of a century ago So things like goiter and cretinism and so forth, which used to sicken millions of people. And at the time, no one knew what caused them, right? But it turns out that for these kinds of diseases, many of them were due to nutritional deficiencies, and we don't understand everything about nutrition. Any of the snacks that were out there at breakfast, you could run them through the mass spec, and you'll find all these chemicals where, to this day, no one has any idea what they do. But that doesn't mean that simple and safe, effective nutritional interventions, like, for example, iodizing salt, haven't been the cure for many of these diseases, which have just vanished. So if you take goiter, you've probably never met anyone who's met anyone who's met anyone who has goiter, right? It's just gone. And so despite the fact that we don't understand the complexity of the microbiome fully, the possibility that there will be safe and effective interventions like that, restoring missing microbes that we know nothing about, in the same way that there were missing nutrients that we knew nothing about a century ago, could be really important as a concept. So this leads to this question of how can we map our microbes, whether we're talking about the scale of our bodies or the scale of our planets. And to address this question, the NIH invested $173 million in the Human Microbiome Project, where we looked at 250 healthy people and sequenced 4.5 trillion bases, so 1,500 times the size of the haploid human genome. And the problem with the data is that it looks like this, right? This is the first 0.1% of the first file of data in the Human Microbiome Project. It's pretty hard to tell who lives where in the environment from this sort of information. You probably can't even tell that that's an oral sample, right? And so what we've been doing is taking this kind of information and turning it into a more interpretable map that you can actually use. And so when we apply tools that we developed in my lab and apply it to the same data set, we get this picture. And so I know what you're thinking. You're thinking, hey, well, I don't understand that either. But just bear with me for a moment. Each point on this map represents all the complexity of a single microbial sample, boiled down to just one single point on that landscape. And essentially, 2 points are placed closer together if they have more similar microbes. They're placed further apart if they have more dissimilar microbes, as read out by their DNA. And you can see some patterns emerging, almost like continents on this map. So you can see that the mouth is very different from the skin, very different from the vaginal community, very different from the fecal community. The algorithm that does this knows nothing about the samples, knows nothing about the people. All it knows about are the similarities and differences in the samples as read out by the microbial DNA. So this means that different parts of the body are really different. And so if I highlight the mouth and the gut of one person in the HMP, you see they're in really different parts of the map. But it was only later when Jack and I, together with Janet Jensen, did the Earth Microbiome Project, that we could really understand these differences, because we could go out on the planet and ask what two communities are as far apart from one another out there in the environment as the mouth and the gut of this one person. So which ones are the same distance apart in terms of their microbial DNA? So if you think of your mouth as being more or less like a coral reef, complex mineralized structures covered with biofilms, maybe your dentist pesters you about those biofilms from time to time, then the amazing fact is that the mouth is as far away from the gut in terms of its microbiology as this coral reef is from this prairie in the middle of Nebraska, right? So this is incredible, because it means a few feet along the length of your body makes as much of a difference to your microbial communities as thousands of miles across the Earth's surface. They're completely different communities in different parts of your body. So this may lead you to wonder, well, where do our microbes come from in the first place? And if you have dogs or kids, as I do, you probably have some dark suspicions about that. All of which are true, by the way. So just like I can match you to your computer equipment by the microbes you leave behind, I can match you up to your dog by the microbes you share. And one thing that's fascinating is that depending on where you live, dogs can actually be a major source of microbial exposure. So in the Midwest, for example, during the summer, there's 3 roughly equal sources of exposure from leaves, from soil, and then from animal feces. But in the winter, there's no leaves on the trees, and the soil's all frozen, so it's not volatile. And essentially what's left at face level, if you're walking around the streets of Cleveland or Detroit, is this dog fecal signal. So that's what you're breathing in. However, the news is not all bad, because as Erica will tell you more, exposure to pets, especially early in life, can be beneficial rather than harmful, especially in terms of controlling allergies and atopic disease. But in all seriousness, how we're born has a tremendous impact on our first microbes as babies. So this is work we did with Maria Gloria Dominguez at NYU a few years ago, looking at mothers an hour before they gave birth, and then their babies within 20 minutes of being born. And what you can see is that, that in babies, unlike the highly differentiated communities in adults, all their microbes look the same. So in pink, we have samples from all over the bodies of babies who were delivered vaginally, and then in red, the vaginal communities of all of the mothers. Whereas over here in light blue, we have samples from all over the bodies of babies who were delivered by C-section. And in dark blue, we have the skin of the mothers. And so we think these differences, these profound differences in the initial microbiome, might be involved in some of the differences in health on average between C-section and vaginally delivered babies. Where if you're delivered by C-section, there's higher rates of asthma, of allergies, of atopic disease, and some studies have even reported a twofold increase in the relative risk of autism. Now, it's important to remember that if you're delivered by C-section, the most likely outcome is that you'll be fine. We're talking about small risks here for the most part, but over a large population, they can really add up. And so in later work with Gloria, we wanted to see if we could actually modify the microbiome directly. So she recruited 3 groups of women, those giving birth vaginally, those planning a C-section with no medical indication, and those planning a C-section but willing to incubate in their birth canal for an hour some sterile gauze, and then coat the baby thoroughly with the vaginal microbes that they would have got anyway had they been delivered naturally. And this came out in Nature Medicine earlier this year. And fascinatingly, what we were able to show is that for a range of taxa and for a range of body sites, a month later, the microbiome of the baby was still profoundly different and resembled the vaginal birth rather than resembling the C-section birth. So, so this was too small a sample to look at clinical endpoints, but we have another 14,000 samples in the freezer on this project that we're just starting to sequence now. And that'll really let us tell not just can we modify the microbiome, which we can, but can we actually impact the child's health by transferring those natural vaginal microbes. So that's what happens initially. You might wonder what happens after that. This is what we did with Ruth Lay, looking at one infant over the first 2.5 years of life, just looking at the fecal samples, and you can see that they start off in the vaginal region of the plot, as we'd expect from the kid's delivery mode. The question is, over 2.5 years, how much do they come to resemble the fecal state? And is that a smooth progression? Are there any events along the way as he journeys through this map and so on? And in case you're wondering why 2.5 years, that's when he was toilet trained. And it's a lot easier to get the sample out of diapers than it is to fight your kid for them after they've proudly learned how to flush. So let's start this going. And what you can see is sometimes there's a lot of change 1 week to the next. Sometimes there's just a little change. Remember that what matters on this map is how far apart 2 points are. So your kid really does look like a different person 1 week to the next, at least in terms of their microbes, because these distances are sometimes larger than the distances between any 2 adult fecal samples in the entire HMP. So coming up here is something fascinating. So he's going to get antibiotics for an ear infection. You see this tremendous regression of the microbiome and doing months of development, followed by a relatively fast recovery. So that went by pretty fast, so I'm just going to rewind it for you and play it for you again. What we see is on administration of oral amoxicillin, tremendous regression of the microbiome, followed by a rapid recovery. And then by the time he gets to 2.5 years, you can see he's basically in the healthy adult state. But that doesn't always happen, and early life antibiotics can sometimes lead to adverse outcomes, including obesity, as shown by Marty Blazer, and even neurocognitive outcomes. And so figuring out how to restore these microbes lost by antibiotics or by C-section or by other treatments is something that we should really be thinking about. So a lot of what I've shown you has been based on community patterns, and you might be wondering, is there any molecular mechanism underpinning this? And this is something we're starting to work on with Peter Dorrestein, also part of the Center for Microbiome Innovation at UCSD. And we're going to switch from the gut to the skin, which provides a really important interface between us and the environments that we inhabit. And the reason for that will become apparent soon, because Peter was interested in sampling himself at 500 sites on his body, and also sampling his wife, Kathleen. And it's a lot easier to do that on the skin than it is, for example, in the gut. And so basically Peter ran this through 2 kinds of mass spec. I did sequencing on the bacteria to figure out what the communities were. And so what's cool about this, basically these diagrams show in dark blue means nothing of a particular compound. Then the brighter the color up through red, the more there is. And so some molecules are broadly distributed on the body, whereas others are localized to the armpit, the groin, or wherever. We can see the same for the microbes, where, for example, staph is in the moist areas like you'd expect, the feet and the nose, propionibacterium on the head and shoulder, carinobacterium over the whole body. And so what we can do is we can then match up the molecules and the microbes. So if you look at propionibacterium on the head and shoulder, you can see oleic acid on the head and also on the hands. And then it only gets oxidized where propionibacterium lives on the head, not where it doesn't live on the hands. So we can guess that that microbe does that reaction. And then the cool thing is we can test that hypothesis in the lab by asking directly if you incubate the bacterium and the precursor, do you get the metabolic product? And so we can develop this into a high throughput assay for understanding which microbes do what in environmental samples. And we can apply this in the built environment as well. So this is Peter's office, for example, where we did a 3D scan of the office using a little LiDAR unit on, that you can stick on an iPad. And again, we can swab everything and match up the microbes and the metabolites. So what's cool about this is that we can use source tracking, for example, to find all the metabolites that are specific to me. So I'm sitting here, you can see a bunch of my metabolites on my laptop and my phone, but nowhere else in the office. We can also do some interpretation of some of these. So you can see this really bright metabolite here on the hands and the mouths of 3 out of the 4 people sitting around this office. And what that molecule is caffeine. And Peter doesn't drink coffee, but the rest of us do. You can also see that some previous occupant of his office has spilled some on the floor. Well, this is definitely the most expensive way to find this out. It gives you an idea of the power of the technique. And we can also apply this stuff in kitchens, for example. So this is work with Noah Ferrer a few years ago, basically tracking different potential pathogens through the kitchen, where they all have different spatial distributions. And then we can track the sources to the palm of the hand, or to the produce, which is mostly on the counters, or the faucet water, which is mostly around the tap. And we can do the same sort of thing in the bathroom. So when we looked at the bathrooms at the University of Colorado, where I was at the time, you can see the skin signal mostly on things you touch, including the toilet seat, which of course you're touching with the skin of your butt. You can see the stool, mostly confined to the toilet stall. And then you can see the dirt, mostly on the floor, but you can also see some on the flush handle. And so apparently in the US, a number of people use their feet to flush. Coming from New Zealand, I had never heard of this practice, but we can pick it up pretty clearly just by looking at the microbiology. And then Jack will tell you more about applying these techniques in the hospital setting and in the home setting, where fascinatingly the patterns are strikingly different from one another. So this might lead you to wonder, well, hey, where am I on this map? And in the Human Microbiome Project, for all its power, one drawback was that there was no way for ordinary people to participate. So a couple of years ago, we launched this project called American Gut, a citizen science initiative that allows you to claim a place for yourself on the microbial map. And Justine and Embriette from my lab are here with kits, if any of you are interested in getting swabbed. It turns out not everyone wants to know what's in there. So these are middle schoolers touring our lab and being informed that we're going to use lasers and robots to look at the bacteria in their poop, all of which is literally true. But it's one of the largest, but a lot of people have been interested. And at this point, it's one of the largest crowdsourced, crowdfunded citizen science initiatives that exists. We've raised over $1 million. We've released data for thousands of samples, including hundreds of samples from PGP participants, which are all linked together with the rest of your profile in Open Humans. And being able to link up the genome to the microbiome, as you've heard from Mike and from others, is incredibly powerful. But we still need more to be able to get good data from those associations between the genome and the microbiome. And so clearly many of you agree, because the American Gut Project was a recipient of the Getty People's Choice Award last night. And this is Embriette if you want to track her down and ask for a kit if you don't have one already and you're interested in sampling yourself. So thank you for that. And on a project of this scale, looking at thousands of people, we see some expected results. So you probably expected that your microbial diversity depends on your age. You might not have expected that it depends on how many hours sleep you get at night, which has just as statistically significant an effect. And then when we do these power curves that Justine devised, so basically a steeper curve means a larger effect. You can see that age and inflammatory bowel disease and antibiotic use are all up there. But one thing that has an effect even stronger than those is how many different kinds of plants you eat. And so it's amazing that this kind of lifestyle factor can have as strong an effect as disease or drugs, and especially because it's something that you have control over and something that you can modify. But we can also tell what sex you are, how fat you are, how much you drink, how much you exercise, or rather how much you say you exercise, because remember, this is all self-reported data, which is one of the limitations of citizen science. But we're expanding this in all kinds of different ways, including initially expanding to British gut, because it's easy to translate the materials from English into English, but also in things like ICU patients, autism, depression, and then at UCSD, some of the sickest people in the cardiac and in the oncology wards, and some of the healthiest, like our student athletes in our healthy aging cohort. And so the most compelling case we have about why you care about this is with Clostridium difficile infection, where C. diff kills 14,000 people a year in the US alone. This is just to reorient you. The stool of C. diff people looks nothing like healthy stool. And so that's these orange spikes here. And so the question is, if you do a fecal transplant, and so what's going to happen is that 4 of these patients are going to get a fecal transplant from the same donor. This is with Mike Sadowski and Alex Kreutz at the University of Minnesota. The question is, like it was for the infantile series, how much do they come to resemble healthy stool, and how long does it take, and is it a smooth progression? But now, instead of being 1 week for each frame, it's just going to be 1 day for each frame. And these patients who are going to the bathroom, in some cases, literally dozens of times a day. And the answer is, with fecal transplantation, immediately you see their whole community state in just a few days going into this healthy region, or their diarrheal symptoms vanish. And people who have been bedridden for months are getting up and walking around. The last large-scale trial that directly compared fecal transplant to antibiotics for recurrent C. diff had to be ended early because the fecal transplant was more than 90% effective, which is typical. The antibiotics were less than 30% effective, which is also typical. And it was considered unethical to continue withholding the fecal transplant from the people on the antibiotic arm of the trial. So the challenge facing us at the moment is to figure out for what other diseases can we identify some kind of dysbiosis, whether it's obvious like this or more subtle like it is for other diseases, and everything ranging from inflammatory bowel disease to rheumatoid arthritis, to even, if the mouse models hold up, things like depression, Parkinson's, and autism, and figure out how to reshape the communities back into the healthy state, whether it's with fecal transplant or with prebiotics or probiotics or more targeted antibiotics or phage therapy or immunotherapy or all of these other things guided by the microbiome. But what we need to do is we need to go beyond this map, because we're pretty good at telling you where you are on this map, and develop more of a microbial GPS that tells you not just where are you right now, but where do you want to go on the map, and what do you need to do 10 by 10 in order to get there. And ideally, we need to develop the system, make it so easy to use that even our children can use it. So you can imagine a kind of smart toilet where as you flush, it does a microbiome or metabolome analysis. It delivers it to your smartphone, which, let's face it, I bet you're using in there anyway, and it integrates it into this kind of animation that tells you, did you go to a good place or a bad place? Maybe gives you some hints about what you need to do to optimize your microbiome for health over the course of your life.